Hello, hi, a very happy good evening. So this is Krishna Ma'am. We're talking about the story of life on earth, and that's biology. So thank you so much for joining my new beginning series. So we are here starting a new chapter. So we have already started a new chapter on Wednesday. So that is, we started off with human reproduction. So today we are going to continue from there. So today we are going to finish off the male reproductive system. So we started off with the male reproductive system. We are supposed to finish off the male reproductive system today. So that is our task at hand. So yeah, so I guess your NEET exams are nearing, and it's time that you guys take up a bit of seriousness and you start preparing, because I don't think competitive exams are very easy, and it is easy provided you put in a lot of efforts to do the best. Alright, so let's continue with our topic about your human reproduction. Hi Veda, yes, I hope you're doing good. So thank you so much for joining my lecture today. So please don't forget to like, share and subscribe. So let's move forward. So human reproduction part 2. So in the last lecture, so we were talking about what are the events in the human reproduction. So these were the topics that we are supposed to discuss. So I told you what are the events in human reproduction. So that was the first thing that we started talking, and then we went on to talk about the scrotal sac or the scrotum. Right, so before we go into the scrotum and the scrotal sac out of nowhere, so I'll give you a link as to what is the scrotum. So before that, let me explain you the events in human reproduction. So events in reproduction. So you have your first thing, human reproduction is sexual. So we follow sexual mode of reproduction. And humans are also viviparous, they give birth to young ones. So the first step is gametogenesis. That is process of formation of gametes, that is your gametogenesis, right? So in case of males, you have the spermatogenesis. In case of female, so we have the oogenesis. So first two different gametes, so fusion of two different gametes is your sexual reproduction, right? Yes, it is biparental. You're absolutely right, Veda. So first your gametes have to be formed. Once the gamete has been formed, the gametes have to meet each other. So this process is insemination. So the male gametes are transferred to the female genital tract so that there is meeting. So once they both meet, then there is fusion. So fusion of male and female gamete, and that is fertilization. So fertilization leads to the formation of zygote. So once the zygote is formed, the zygote undergoes a series of division. So if this is the zygote, so this undergoes series of division like this. So this is cleavage, series of division is cleavage, and finally it ends in a particular 8 to 16 cell stage, that is blastocyst stage. Alright, so after blastocyst you have implantation of the zygote on the uterine wall. So after implantation you have the embryonic development. So after the embryonic development, excuse me, so actually the embryonic development is your gestation. So it takes 9 months in humans. After that you have the childbirth happening. So childbirth is parturition. So parturition is childbirth, right, followed by lactation. So these are the stages in human reproduction. So before we actually start talking about gametogenesis, where the male and the female gametes are produced, we started talking about the male reproductive system because we have to know what produces the gametes, right? So what is the design of the reproductive system? So we started talking about the male reproductive system. Right, so this male reproductive system, so this includes your pair of testes, So then you have accessory glands. Then you have accessory ducts, and then external male genitalia. So these are the 4 members of your male reproductive system. So we started talking about your testes in the previous class. Yes ma'am, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. So pair of testes. So we know testis is located outside the abdominal cavity. So why it is located outside the abdominal cavity? Because to produce sperms, it needs a much lower temperature. So that is why it is situated outside the abdominal cavity. So you have a pair of testes, and the testis is suspended inside a particular sac that is called a scrotum or scrotal sac. Right, so this is what we were discussing in the previous lecture. So I hope you got a connect between what I spoke last. Okay, so here we look into it. So here you have the scrotal sac. So this is the testis, right? So then it is suspended inside your scrotum. So scrotum is actually made up of 2 types of muscles, if you remember. So it is made up of your dartos muscle. So dartos muscle is responsible for regulating the temperature. And it is made up of spermatic fascia. So fascia is your connective tissue, right? So this is what lines your scrotum, right? So your scrotum is something like this, and your testis is like this. So your scrotum is attached to the wall of the abdominal cavity through what is called as inguinal canals. So you have the inguinal canal. It is through the inguinal canal. It is attached to the abdominal wall. Okay. So your testis is attached to the scrotum by a thin fibrous connective tissue that is called as gubernaculum. Okay, understood? So are the terms that I have mentioned clear? So now let's move further. So spermatic cord, okay, so we were talking about it. So we have your vas deferens, gonadal veins and gonadal arteries. So these are the structures through which blood comes in and all the functions happen, right? So spermatic cord actually is one which connects your scrotum to your abdominal wall along with your dartos muscle, right? So here you have the vas deferens. So you have the gonadal veins and the gonadal arteries. Spermatic cord is a bundle, right? So inside that bundle you have what is called as vas deferens, you have gonadal veins, you have gonadal arteries, and you have your nerves, you have your lymphatics, right? So all these has to pass through your testis, right? So during the embryonic stage, the testis is in the abdominal cavity only. So when the child is born, only then the testis descends down into the scrotum. So in the starting, when the embryo is developing inside the mother's womb, the testis is in the abdominal cavity only. At the time of birth, it descends down into the scrotum, all right? So at the time of birth, it descends down to the scrotum. If it fails to happen so, if the testis fails to descend down into the scrotum, so that condition is called as undescended testis or cryptorchidism. So then doctors can surgically pull it down to the scrotum, or you can give hormonal injection. So orchiopexy, where undescended testis is surgically moved into the scrotum, where the doctors bring the testis into the scrotum if they do not descend. Alright, so the internal lining of testis has 3 coats. So now we are coming to the portion inside your testis. Alright, so this has your tunica vaginalis, so tunica albuginea and tunica vasculosa. So we have 3 layers inside your testis. So long we were talking about the pouch that encloses your testis. Now talking about your testis, so that has 3 coats. So you have your tunica vaginalis, tunica albuginea, tunica vasculosa. So tunica vasculosa is the innermost layer. Right, so tunica vaginalis, so this is the outermost, so this is parietal, so on the periphery, so this is a visceral layer. So it covers the whole testis except at the posterior border where the vessels and the nerves enter. So basically if a testis is there, so it covers the entire testis like this except at the posterior point where the blood vessels and the nerves enter the testis. So tunica albuginea, it's a dense white fibrous coat. It has a posterior border that is thickened. So towards the posterior side it is thickened, and it has a vertical septum, that is the wall, that is your mediastinum testis. So don't worry much about it, just remember the 3 layers of your testis if you are very confused. So that will do all the good. Right, so this is what I was talking about. So here can you see the mediastinum? So it forms a septum. So here I am talking about the 3 layers inside the testis. That's all. Okay. So now explaining the structure of testis to you. So the testis, so we have a pair of testis. So the testis is divided into a huge compartments, into 250 compartments, which is called as your testicular lobules. So inside each lobule you have 1 to 3 highly coiled structure. So this 1 to 3 highly coiled structure is called as your seminiferous tubules. Okay, so this seminiferous tubules have 2 types of cells. So one is your male germ cell, the other one is your Sertoli cells. So male germ cell undergoes spermatogenesis to form sperms, and the Sertoli cells nourishes the male germ cells. Right, this is the function of your seminiferous tubules. All right, yes. So when I say I have huge compartment, so say this is one particular compartment, and inside this I have highly coiled structure like this. So this is my seminiferous tubule. The space in between the seminiferous tubule, the space in between the seminiferous tubules, so that is called as the interstitial space. So this interstitial space has interstitial cells. So this interstitial cells are your Leydig cells. So if you remember when we were talking about chemical coordination and integration, so your LH, that is your luteinizing hormone, acts on your LH to form a group of androgens, that is our testosterones, right? So that is this Leydig cell. Fine, clear? Yes, did you understand the design of testis? So testis is oval shape. So the length of the testis is 4 to 5 centimeter in length and 2 to 3 centimeter in width. So it is covered by a dense covering. So each testis has 250 compartments, that is your testicular lobules. Alright, so each lobule has 1 to 3 highly coiled seminiferous tubules. So that is where your sperms are formed. Hi Pranav, so thank you so much for joining my lecture. So please don't forget to like, share and subscribe. So each lobule has your highly coiled seminiferous tubules, and inside the seminiferous tubules you have 2 cells. One is the male germ cells, which is also called the spermatogonia, which undergoes spermatogenesis to form your sperms. The other one is your Sertoli cells. The other names of the Sertoli cells is your subtenacular or the nurse cells. So this nourishes your sperm cells. right? So this seminiferous tubules are lined by white fibrous connective tissue. So CT in my slide is connective tissue. So that connective tissue is tunica propria. So that is a type layer of connective tissue. So on the inner side, it is lined by your cuboidal germinal epithelium. So this is the structure of your seminiferous tubules. So outside the seminiferous tubules, there are interstitial spaces which contain small blood vessels, interstitial cells called the Leydig cells, and immunologically competent cells, that is your antibodies. So all these are present in the space between the seminiferous tubules. So Leydig cells synthesize and secrete testicular hormones, that is your androgens. So testis acts on the testicular lobules. So now it moves inside the seminiferous tubules. They join at the apices of the lobules to form the straight tubules called as the tubuli erecti. So this is my seminiferous tubule. So 1 to 3 highly coiled structure is my seminiferous tubule. Right? So now all the apices join like this. So all the three join together at one point. So that is my tubuli erecti. Okay. So now it enters the mediastinum, that is the innermost, the second most layer. So that is my rete testis. So if you can see, so they all join together and then enter the mediastinum. So this is one testicular lobule, this is one lobule, this is another one, and this is another one. So all the apices of the seminiferous tubules join together to form the tubuli erecti. Then they open into your rete testis. Alright. So with this, we are done with the structure of testis. Now we are moving into male sex accessory ducts. So what are they? So you have the rete testis, vasa efferentia, epididymis, and your vas deferens. So from the testis, so inside the testis, you have the seminiferous tubules and they join like this. This is your tubuli erecti. And then they enter the mediastinum, right? So this is our tubuli erecti. Then they open into the rete testis. So from the rete testis, it moves into vasa efferentia. Then into epididymis. Then into vas deferens. This is the order in which it reaches the abdominal cavity. So see, so testis is descended like this. So it has to move like this, right? So you would have seen a structure like this. So the way through it reaches the abdominal cavity is this passage. So testis is here, so rete testis is here, vasa efferentia, epididymis, vas deferens, and then it loops over the urinary bladder. So seminiferous tubules in your testis opens into rete testis, this opens into vasa efferentia, then the epididymis, then the vas deferens. So vas deferens ascends to the abdomen and it loops over your urinary bladder. So UB is the urinary bladder. So seminal vesicle has a duct. So when it reaches the urinary bladder, so this shape is your urinary bladder. So from here it reaches the urinary bladder. So from the vas deferens loops over the urinary bladder, right? So here there is a group of glands, a pair of glands called the seminal vesicle. A gland will have a duct. So the duct is called as the ejaculatory duct, and the base of the ejaculatory duct is swollen. So that actually stores and transports the sperm from the testis to the urethra. So urethra is the final opening, right? So the urethra originates from the urinary bladder, right, and it extends till the penis. So penis is the external genitalia of the male to its external opening, that is urethral meatus. All right. So this is the external male genitalia. So that is your penis. So hi Sumila, hello. So thank you so much for joining my session. So please do like, share and subscribe. So I hope you guys have understood. So first it is the seminiferous tubules of the testis. Then you have the rete testis. From the rete testis it moves into your vas efferentia. Then you have your epididymis. Then you have your vas deferens. Alright. Fine. So from the vas deferens, what happens? It moves along with the ejaculatory duct. It is a duct of your seminal vesicle and it comes out through the urethra and finally through the external male genitalia, that is your penis. So it is made up of a special type of tissue. So this tissue helps in the erection of penis, so that facilitates insemination. So insemination is the process by which your male and the female gametes meet, right? So this erection of tissue that is present in your male external genitalia does it. Alright, so the enlarged end of this penis, that is the external genitalia, is glans penis and it is made up of a loose fold of skin that is called as foreskin. Alright, so with this we are done with the external genitalia as well. So now talking about the male accessory glands. Hi Abhirami, hello. So thank you so much for joining my lecture. So I hope you're enjoying. So please don't forget to like, share and subscribe. So now we are talking about the glands. So we are done with the pair of testes, the ducts as well as the external male genitalia. So now we are going to talk about the accessory reproductive glands. So we have three glands here. So seminal vesicle, you have prostrate glands and you have bulbourethral gland, right? So first we will be talking about the seminal gland. So it is lined by glandular epithelium and it secretes the seminal fluid. Alright, so the fluid is lubricating, meaning it helps in movement. It is transparent and it is jelly-like in the sense that 70 to 60% are semen. So the pH of this fluid secreted by the seminal vesicle is 7.3, right? So fructose is present inside this fuel that acts as a fructose is present inside the secretion of the seminal vesicle and it acts as a fuel to your sperms, meaning your sperms move with the help of fructose. So it also has your fibrinogen, your prostaglandin and citrate inositol, so which enables your sperm to move inside your female genital tract. Alright. So next you have the prostrate gland. So they are located below the urinary bladder. So prostrate gland is single, they are not paired. Only your seminal vesicle and the bulbourethral gland are paired. Okay. So this has a slightly alkaline fluid. It is milky, sticky, jelly-like. Alright. So it makes 30% of the semen. So it makes the activated semen. Alright. Yes. Yeah. Very sticky. So secretions of the prostrate gland plus the seminal vesicle makes the semen coagulate, meaning it slows down the speed of the semen because it has to conserve the energy, right? So that is the reason. So coagulated semen reduces the energy so that it does not have to use a lot of energy inside the male reproductive tract itself. So that is why it coagulates. Fine. So the last one here is your Cowper's gland. The Cowper's gland are also known as bulbourethral gland. So they are a pair of glands. So they are located on the either side of the urethra. Lateral means on the sides of the urethra. They secrete transparent, slimy, jelly-like fluid. Alright. So here the pH is 7.2. It destroys the acidity of the urethra and cleans it for the movement of sperms. If you see, there is only one passage for the secretion, ejaculation of sperms. Okay. Coagulation means very, very thick and viscous. So if I say fluid, it is watery. Coagulation means it forms clots. It is not very fluid. It is very, very viscous and very dense. It does not allow rapid movement. That is coagulation. Alright. So if you see, there is only one passage for excretion of urine and for the ejaculation of semen, right? So for the ejaculation of semen, we need an alkaline pH, and for the micturition process, that is when you release urine, you need an acidic pH. So you have the bulbourethral gland which actually clears off the acidic pH so that the semen can move through and the sperms can be ejaculated. So with this, we are done with the male reproductive gland. So on Monday I'll be starting with the female reproductive gland. So please join my lecture on Monday. Yes, Sumila, that's fine. So thank you. You have given me an opportunity to clear your doubt. So thank you. So it's nice to see you guys here, so active and actively texting. So very proactive, I would say. So thank you. So we will stop here. So if you genuinely liked my way of teaching, so please do like, share and subscribe. So please tell your friends, your juniors, etc. So thank you. So stay tuned and have an amazing weekend. I will meet you on Monday. So until then, bye and take care.